traveling through another dimension. A dimension not only of sight and sound, but of mind. A journey into a wondrous land whose boundaries are that of imagination. Your next stop, the Twilight Zone.
Listeria monocytogenes is a oxidase negative, catalase positive, and gram-positive rod-shaped bacterium. It can be arranged into short, chain-like structures. Longer cells may resemble coronate bacteria, and the smears can result in Listeria tulacocoid. Listeria monocytogenes bacterium grow well on cultivation media. On blood agar, it forms small colonies about 1 to 2 millimeters in diameter after 24 hours of cell growth. Listeria monocytogene colonies are beta homolytic. The Bacillus subtilis has the closest evolutionary relationship with Listeria. Listeria is a highly diverse species and has at least 2 distinct phylogenetic lineages with differences in their evolutionary history, population structure, and horizontal gene transfer. One line of descent is the Listeria monocytogenes species group. Others include Listeria roquefortii and Listeria fleishmani. The phylogenetic interrelationships of the genus Listeria were studied by sequencing of 16S rRNA and the HLY-A gene. In 1924, Murray isolated the bacterium from laboratory animals and classified the gram-positive Listeria bacterium as the new agent bacterium monocytogenes. In 1940, James Hunter Harvey Peary named the genus Listeria after Joseph Lister. Roquefort separated the Listeria monocytogenes from other Listeria species using biochemical methods and genetic comparisons. Big outbreaks of Listeria in history include the outbreak in 1981 in Nova Scotia, and more recently in 2015, it was found in the Bluebell creameries. Listeria can enter the food chain at any point. Listeria is commonly found in soil, stream water, sewage, plants, as well as in food. It can be found in avians, mammals, aquatic animals, and humans and animals may consume them. Listeria is known to colonize surfaces. Listeria biofilm formation can be impacted by the environment within food processing facilities, which would trickle down to the consumers, such as livestock and humans. Listeria is mainly transmitted via foodborne routes. Furthermore, Listeria can also survive in many extreme conditions, such as high salt concentrations, high pH, high temperature, and temperature as low as 4 degrees Celsius. Listeria are microaerophilic facultative anaerobes able to survive with or without oxygen in its environment. Listeria can produce adenosine triphosphate through a complete respiratory chain and also have several fermentation pathways, including heterolactic fermentation and homofermentation. Listeria enter the host through their GI tract but can infect the monocytes, macrophages, and polymorphonuclear leukocytes. It is a pathogen that lives inside the host cells to survive. Pathogenic Listeria go through an intracellular life cycle by escaping from the phagocytic vacuole, rapid intracytoplasmic multiplication, and directly spread to its neighboring cells where they repeat the cycle. Listeria bacterium is first phagocytosed by cells and secrete a pore-forming toxin called listeriolysin. This enables the bacterium to escape from the phagosome. At this point, the bacterium go through rapid division in the cytoplasm while evading immune response and moving throughout the cytoplasm from cell to cell using the actin motility. Listeria express internalin A and internalin B, which induces bacterial internalization into non-phagocytic cells. After the Listeria bacterium is phagocytosed, it secretes a pore forming toxin called Listeria lysine, which helps it to exit the phagosome to the cytoplasm, where it can reproduce. ACT-A protein induces actin polymerization on the bacterial surface and creates a tail to help motility. Listeria is an endosymbiont. The interaction between Listeria and its host is parasitism. Listeria invades and survives within the host cell by utilizing their host's cellular machinery. The host is at a disadvantage in the relationship. Listeria monocytogenes is a pathogenic species of Listeriae. Listeria causes listeriosis in animals and humans and found to be widely distributed in nature. Listeriosis is an infection caused by eating food contaminated with the Listeria bacteria. Listeriae has been isolated from crustaceans, fish, oysters, ticks, 42 species of wild and domestic animals, and 17 avian species. Listeriosis is common in cattle, sheep, goats, and pigs. These grazing animals ingest Listeria and contaminate vegetation and soil. The animal-to-animal transmissions occur by fecal to oral route. In the United States, listeriosis has been recognized as an important public health issue. The disease can have a devastating impact on pregnant women, newborns, and adults with weakened immune systems. The Listeria disease overt form has a mortality greater than 25%.